Welcome to Snow Lecture 18 of Molecular Biology, 27th of February 2019. Since the snow is actually still falling and was falling quite a bit at my house this morning. So as I mentioned on D2L, the material that we cover today will be on the final exam. Before that, I have a couple of other quick announcements. Speaking of exams, a couple of people who did very well on the second exam, particularly those who improved very well between the first exam and the second exam, have sent me a couple of study hints, and I've posted all of those in the study group section in D2L. So go to the study group section, look that up. You'll find a couple of hints on how to do well on future exams. So with that being said, today we're going to continue talking about translation. Last time we talked about the genetic code, open reading frames, tRNAs, tRNA synthetases, and a little bit about the ribosome. We're going to pick up on the ribosome and then talk about translational initiation and quality control. Folding we'll actually talk about on Friday. Hopefully we'll actually have a real class then. Reminder, any more questions come up, please feel free to send me an email, and I will post answers to all of your questions on D2L. So last time we looked at the ribosome cycle. What happens when you have ribosomal, you have a messenger RNA that's being produced by an RNA polymerase. This could be bacterial RNA polymerase, could be eukaryotic RNA polymerase. First thing that happens is you have the small subunit and the initiator tRNA associate with the start codon, AUG in eukaryotes, usually AUG in bacteria and archaea. Then you have the large ribosomal subunit that associates. Then you have an elongation process that requires your aminoacyl tRNA. Transpeptidation happens, translocation of the polymerase, and eventually reaches a stop codon in red. The polymerase, the amino acid polymerase, also known as the ribosome, will dissociate from the RNA at this point. The protein will be released, the RNA will be released, and this whole process can go around again. As I mentioned last time, the ribosome is really all about the RNA, and this is the structure of all of the RNA with one tiny little protein here. That is the large subunit of the ribosome. The small subunit of the ribosome is also very similar in terms of mostly being RNA. There are definitely proteins that are involved as well. And all of these RNAs, actually even in different domains, separate structures. So again, just like we have in proteins, we have a separate domain of RNA that's associated with the ribosome. So this bit of RNA folds up together, forms a particular domain, actually has a particular structure. This one folds up, has a particular structure, and so on and so forth. And it turns out you can actually take these individual domains and put them together and get a small amount of transpeptidation activity from the ribosome. Where do the tRNAs fit into the ribosome? So now we're looking at both the large and the small subunit of the ribosome. Basically, you take this one and flip it over 90 degrees, stacks on top here. These three red, orange, and yellow colors are the tRNAs that fit inside the ribosome, again, between the large and small subunits, and the messenger RNA will be right here, going in between the small and large subunits as well. With these three sites that tRNAs bind to in the ribosome, they are called the E site, the P site, and the A site. The E site is the exit site. This is where the tRNA that no longer has any amino acids associated with it, that will now fall out. And that here is in this red position. So that will be the tRNA that is released. The P site, this is known as the peptidyl transfer site, or the peptidyl site. This is where your tRNA, again here in orange, this now has the growing polypeptide chain attached to it. And then we have the A site. This is where the aminoacyl tRNA binds to. And as long as it binds appropriately to the codon, the anticodon-codon interaction happens, then you have transpeptidation that takes place. and then this whole ribosome will move over one. Then you have the tRNA that was in the A site is now in the P site. The tRNA that was in the P site is now in the E site. And the tRNA that was in the E site is now actually released. So that brings us to what I promise is a clicker question for today. How many different binding sites are there for tRNAs in the ribosome? I'll let you think about it for a little bit. And hopefully all of us will have gotten the correct answer, which of course is Stedman's favorite answer, C. So P site, A site, E site in the ribosome. Hopefully this wasn't too ridiculously easy and everyone will complain. There are some really nice translational animations which are available online, and also an interview with Harry Noller. We're going to take a quick look at this first animation right now, and this is also from the DNA Learning Center. So if we look at this,
the job of this mRNA is to carry the genes
animation from the DNA Learning Center. Here we have the RNA coming out of the nucleus, and now we have the ribosome, both large and small subunits, moving along this RNA. All of these small L-shaped objects are the tRNAs, and here is our protein being made amino acid by amino acid from the ribosome. Once it's done, it folds up and falls off. This process starts by the small subunit of the ribosome binding to the 5' end of the messenger RNA, moving along it till you have reached the start site, then the large subunit associates, and then we need to have all of these tRNAs. So the tRNAs here, they've got that little red spot at the end. That little red spot is the amino acid attached through the high energy bond by the amino acyl tRNA synthetase. Now if we pull off the front, and this would be the large subunit of the ribosome, you can see where those tRNAs actually fit into the structure. And it's really tight in between the large and the small subunit of the ribosome. Here again, each of those moving from the A site, P site, E site, and each of these tRNAs comes in with its amino acid that gets attached. This growing polypeptide chain, eventually is going to be the protein, gets outside of the ribosome and is then released. So here again, we've got our A site, our P site, and our E site. P site, where you have the growing polypeptide chain. The A site, where you bring in this new amino acid to add it to your growing chain. So again, looking at it from the side, now we have this A site and all of the tRNAs that are coming into this particular A site. Now, one thing that's not shown here is that, of course, it's not always going to be exactly the right tRNA which gets added. In fact, there are a bunch of tRNAs which are the wrong tRNAs, so it actually takes rather a long time to find the right tRNA that's going to match with your codon in order to get this elongation step to take place. And we'll look at the process of that a little bit more a little bit later on in today's lecture. As the protein is made, you can actually see here that it's sort of forming a helical structure, and then maybe a beta sheet, and then it's folding as it's coming off of the ribosome. The ribosome itself is just making this polypeptide chain, but then the protein will fold up. Once it's folded up, it's complete and is then ready to go. Let's go back to our presentation now. The other interview, which we're not going to go and look at here, is an interview with Harry Noller, who is a professor, I think professor emeritus now, at UC Santa Cruz, has really spent his career studying ribosomes. And because of that, he won this breakthrough prize that some of you may have heard about, sponsored by Google and Facebook and all of those people, for a cool $3 million. So he's quite happy to have gotten that. So now let's look at not just the act of translation itself, we'll take a look at translational initiation. And so this is now a little different between bacteria and eukaryotic systems, but there are a few things that are all the same. What you need to do is get the messenger RNA, your initiator tRNA, and the small subunit of the ribosome into a place where you can have the start codon and the anticodon of the initiator methionine. In bacterial systems, it's a formal methionine. It's actually a modified methionine. And this, as we talked about a little bit in the lecture last time, it's actually a different tRNA than would be used if you had an AUG in the middle of your start codon, in the middle of your open reading frame here, a little bit further along through the process. So base pairing between the start codon, your initiator tRNA, and the small subunit of the RNA, and then, and only then, do you get the association of the large subunit in order to form your final process. And this is partly why I showed you that animation, just to show that where the P site is, the P site is crammed right in the middle between the small subunit and the large subunit of the ribosome. There's actually not enough space to get a new tRNA with an amino acid attached to it into this part of the ribosome. So the initiator tRNA has to associate with the AUG before you have the association of the large subunit, LSU, to the rest of the ribosome. So how does the small subunit and the initiator tRNA know where to start? That's translational initiation. And in bacteria, this is due to sequence-specific interactions between the small subunit of the ribosome, which is mostly RNA, forms base pairs at its 3' end with a sequence at the 5' end of your messenger RNA. As usual, we've got 5' to 3' in this direction, 5' to 3' in this direction, forms base pair interactions. This base pairing interaction is either known as a ribosome binding site, but also a Shine-Dalgarno sequence from the Shine and Dalgarno researchers who discovered this in the very first place. So it's a sequence-specific interaction between the small subunit of the ribosome and the messenger RNA near where your start codon is. This subunit, again, 16S is the RNA, 30S is the whole subunit, again together with all of these proteins. So sequence-specific binding next to a start codon, AUG, but again in bacteria this could be GUG and UUG, but it makes sense that this would be used because it's going to be next to one of these ribosome binding sites. If you look at a typical messenger RNA in bacteria, and we talked about this when we talked about messenger RNA processing before. Unlike what you have in eukaryotes, this has a 5' tar phosphate. There's no cap here. And many of these mRNAs in bacteria are polycisteronic, i.e. they can encode multiple completely different proteins, here shown as alpha, beta, and gamma. Each of these proteins has its own start and stop codon. You could also talk about this as being an open reading frame here, start to stop. And also each of them has a ribosome binding site associated with it. So this ribosome binding site is bound by, again, the small subunit of the ribosome together with base pairing interactions at the 3' end of the 16S ribosomal RNA. And that binding gets the small subunit of the ribosome there. That will allow the initiator tRNA to bind, and large subunit, you can get translation. Once that stops, you have reassociation of the ribosome to one of these ribosome binding sites. That can translate protein B, again comes off, starts again, and goes through this process. This happens while the RNA is being made by the RNA polymerase. So this is a co-transcriptional translation process. As soon as that first ribosome binding site is released from the RNA polymerase, the shine dog RNA sequence will be bound, this ribosome binding site will be bound by the small step of the ribosome, and you'll get translation. So even before you finish transcribing this whole messenger RNA, proteins will already be being translated. So that's how you get the small subunit of the ribosome associated. But there are also a number of these so-called initiation factors, or the IFs, and we'll see lots of IFs. We'll see IFs for initiation factors, EFs for elongation factors, and RFs for release factors, which happens at the end. So factors, of course, these are proteins, and these proteins, IF1, IF2, and IF3, each have separate roles in translational initiation. And when we see eukaryotes, actually they've got the same kinds of functions. So what you have with these three, IF1, IF2, and IF3. IF1 blocks binding of tRNAs to the A site, because until you have the initiator tRNA bound here, you don't want to have any more tRNAs bound here. You also don't want to have the large subunit binding to the small subunit. That's what IF3 does. Most important, however, is IF2. IF2 is a GTPase. In fact, it's a classic G protein, so hydrolyzes GTP, and in that process changes the structure of IF2, and the changing structure of IF2 means it changes its interactions with whatever its partner is, and the partner here is the initiator tRNA. So IF2 brings the initiator tRNA to the P site, again, just in the presence of the 30S subunit of the ribosome. Once it interacts, there's GTP hydrolysis, it's released, and once that is released, IF1 is released, IF3 is released, the large subunit will come and bind, and you can start to get translational initiation. So this is easy, quote unquote, doing air quotes here, sorry you can't see them in this recording, easy in quotes, because you have the specific binding of the ribosome, and IF3, IF1, and IF2 bring in the initiator tRNA, and again, once that's brought in, you have hydrolysis of GTP by IF2, and the large subunit can associate. Eukaryotes, however, are kind of different. And we mentioned this before last time, but this is now looking at this process of where the AUG is in your messenger RNA for a eukaryotic system, because eukaryotes don't actually have this nice base pairing interaction to find a nice ribosome binding site. So where you start translation was actually found a lot like we talked about for promoters earlier, consensus sequences. So consensus sequence here is looking where all of the messenger RNAs start, lining them all up with each other, and then figuring out where your protein starts. And 100% of the time, you've got an A, a U, and a G at your start codons. Turns out that -1 from this, you often have a C or a G, and -3, you very often have an A and sometimes a G or a C. +4, sometimes have a G as well. So actually, it's not just the AUG which is important, but also these flanking nucleotides that are important to make sure that you get translation starting at this particular place. So why is it so important that you get translation starting at this specific place? Well, that's because we have caps. And caps are where eukaryotic ribosomes start to associate with messenger RNA. Hopefully all of us remember the cap binding complex, which is what is helping that messenger RNA to get outside of the nucleus. Once it gets outside of the nucleus, the cap is actually exchanged for this protein called EIF4E. Now, EIF4E is the cap binding protein. EIF4E also associates with EIF4G. Now, E is just eukaryotic, IF is initiation factor, and then just various different numbers. So EIF4E and EIF4G are really confusingly called EIF4F. Both of them together end up being this 4F. Why is that? That's because when they were originally purified, talked about protein purification way back when, I guess lecture 3, protein purification, they just took various fractions from their columns. One of those fractions was EIF4F, turned out to actually have EF4E and 4G in it. So we're just going to talk about E and G. Again, E is the cap binding. EIF4G is actually a really interesting protein. It serves as a bridge between the poly A binding protein at the 3' end of your messenger RNA and the cap, which is present at the 5' end. So what that means is these messenger RNAs are always in circles. The 5' end and the 3' end are always attached to each other. Okay, so this is our messenger RNA. Now we need to get the small subunit of the ribosome in. Now, the small subunit of the ribosome here in eukaryotes is different in that it's also associated with this initiator tRNA. So in the absence of messenger RNA. So in bacteria, you always have messenger RNA, again, through Shine Dog RNA sequence, interacting with the ribosome, then the tRNA comes in. Here, the tRNA is already associated with a small subunit of the ribosome. This complex, also with an initiation factor, also a GTPase, this binds to your circular RNA at the 5' end. After it's bound to the 5' end, then this small subunit of the ribosome actually moves along until it reaches this start code on the AUG, which is going to match now to the anticodon of your initiator tRNA. Once that happens, you have this step. So moving along, and actually moving along, this actually requires ATP hydrolysis. Probably the ATP hydrolysis is mostly for getting rid of secondary structures, i.e. binding by the RNA to itself in this process, in order to translocate this small subunit of the ribosome together with the initiator tRNA to the start codon. And once you have the start codon and the initiator tRNA, now EIF2 will hydrolyze this GTP. Hydrolyzing the GTP allows the association of the large ribosomal subunit. And once you have the large ribosomal subunit bound, now you're ready to go. You can have your aminoacyl tRNA comes into the A site, binds or doesn't bind, as the case may be, to the messenger RNA here. If it's a correct binding, then you end up getting a peptide bond formation. The peptide bond formation between the methionine, which is your start, and the amino acid, which is present here at the A site, where you have base pair interactions, codon-anticodon interactions. This now moves the amino acid, which was attached to the P site tRNA, again, the initiator tRNA in this case, to the amino acid, the N-terminus of this amino acid in the A site. And then this whole process will translocate. This tRNA ends up in the P site, then the A site, this tRNA ends up in the E site, etc. And we'll look at that in a little bit more detail later on. Another way of looking at that, not using your textbook, is looking at where we have EIF4G and EIF4E. EIF4E again binds to the cap structure. EIF4G provides a bridge to the poly binding protein. So you have a looped structure here, now purple messenger RNA. And then the other initiation factors, EIF3, which actually serves a very similar role to IF3, i.e. it's blocking the association of the large subunit of the ribosome, and then EIF2, which actually has multiple proteins here, but it serves the role of getting this initiator tRNA together with the large subunit of the ribosome to first the cap, then we have translocation until we get to the AUG, the start codon. At the start codon, you have association of the large subunit of the ribosome, dissociation of EAF2. EAF2 can come back, find the start again over here, start again, do another process, get another ribosome onto this messenger RNA, and so on and so forth. One of the reasons I wanted to mention this is it turns out the translation is also dysregulated in many, many different cancers. So many cancers will have a lot of EF4G and EF4A, again, EF4F, this complex. You've got lots of this, which means you're going to have more translation. EIF3, if you're lacking EIF3, you're not going to have as good association, or I'll say blocking the association of the large subunit polymerase, so maybe you get more association of the polymerase. EIF5A, which actually helps with this process of getting your initiator tRNA. Sometimes you have more of this. And so this process, this whole process, again, cap binding, translocation, reassociation, and forming the circular RNA, these are dysregulated in many, many cancer steps as well. So let's get back to our textbook version. Now we have done this transpeptidation reactions, and now the whole ribosome is moving along. P site has now growing polypeptide chain associated with it. A site, the newly bound new tRNA that has the appropriate amino acid, which is going to get covalently bound to the amino acid which is present here. So you have this transpeptidation reaction. All of this activity takes place in the RNA of the ribosome. Then we have a translocation step. And that translocation step is where the growing amino acids chain, which is bound to now the tRNA in the A site, that moves over to the P site, etc. Here this is shown as sort of a jumping mechanism. This moves from here to here, and then the small subunit moves after that. It turns out that this is actually more of a ratcheting process. And actually the two subunits, and I'm showing my hands here, sorry you can't see it on the video, that each of the subunits actually turns relative to each other. So it's kind of like doorknobs twisting in different directions in terms of how this elongation process takes place. So after you have the large subunit that moves, then the small subunit moves, now we have a free A site. This free A site then will allow this translocation to take place. So one of the things I mentioned in the video that they didn't show a whole lot of was more of the protein factors that are involved in this process. The first of those that I wanted to talk about were the elongation factors. So remember we had the I factors, that's the initiation factor, IFs. IF2 is the really important one. That's the one which is bringing in the initiator tRNA. IF3, blocking the large subunit of the ribosome. IF1 is blocking the A site. Now we have the E factors, elongation factor G, which is what you have in bacteria. Eukaryotic elongation factor 2. Both of them do basically exactly the same job, which is helping elongation. So this helping of elongation, after you have the extension of this polypeptide, again on the tRNA which is in the A site, this large subunit rotates a little bit relative to the smaller subunit. And then EFG associates with the polymerase. EFG then hydrolyzes GTP. There's a conformational change, and it helps twist the small subunit to the next position. Eukaryotic elongation factor 2 does basically exactly the same thing. So you have this translocation. Translocation can happen in the absence of any of these, but you can also stimulate that by the presence of these elongation factors. So that's helping to move the polymerase. But that doesn't get us to the thing that was really missing in that animation that we just looked at. The main thing there is that most of the tRNAs, assuming that we only have 20 tRNAs, and of course we usually have more than 20 tRNAs, the tRNAs have to find the right codon to anticodon interaction. And so how is that controlled? That's controlled by other elongation factors, EFTU in bacteria, eukaryotic elongation factor 1 in eukaryotes. So you have a ribosome, you've got your P site with the growing polypeptide chain on it, the E site with the tRNA that doesn't have anything attached to it anymore, and this open A site. This open A site has a codon in it. Now, what has to happen is the cell needs to go through all of the tRNAs with anticodons that are going to bind to this codon. If you have an association of one of these anticodons to a codon that is incorrect, then the tRNA comes in, the tRNA comes off. If, however, you have the correct interaction of a anticodon-codon bases, then EFTU, or eukaryotic elongation factor 1, hydrolyzes the GTP and dissociates. Now you have a tRNA in the A site with the appropriate amino acid that can now be transpeptidase activity, elongating this chain, and then EFG, or eukaryotic elongation factor 2, will help move the ribosome along so you can move to the next step. So lots of GTP, but also ATP required. So that brings us to our next clicker question, which is, what seems to be the main role for ATP hydrolysis in translational initiation by eukaryotic ribosomes? Ribosome translocation from codon to codon, removal of secondary structure in messenger RNA, checking to make sure codon-anticodon pairing is correct, coupling to transpeptidation, or association of the large and small subunits of the ribosome. Let you think about this for a second. So ribosome translocation from codon to codon, that's EFG, or eukaryotic elongation factor 2, GTPase. Checking to make sure codon-anticodon pairing is correct, that's EFTU, or eukaryotic elongation factor 1, also a GTPase. Coupling to transpeptidation. Transpeptidation is happening because you already have a high-energy bond made by the amino acyl tRNA synthetase, originally from ATP, but that's your amino acyl tRNA, so you already have that before you have any kind of translational initiation. Association of large and small subunits of the ribosome, that's actually blocked by initiation factor 3, or eukaryotic initiation factor 3. So that only leaves us with removal of secondary structure in your messenger RNA. So the correct answer to this clicker question is B. Now let's take a look at what's actually known about these structures. I showed a bit of a cartoon version, but it turns out that there are actually some really nice high-resolution structures of this process, and particularly the elongation process, where we have EFTU and the tRNA present here in the A site. So E site tRNA in yellow, P site tRNA in green, and this EFTU in red, the tRNA in the A site here in purple. So quick aside here, where the actual peptide transfer takes place is known here as the peptide transfer center. That's right here in the middle of the large subunit of the ribosome, which is this orange version up here with the proteins here in dark. Small subunit is in light blue for the RNA with these dark blue proteins that are associated with it. And down here is just a zoom in to this particular region here, right at the bottom of your tRNA in the A site. You can also see the messenger RNA here, which is in blue. That then is coordinated with EFTU, which actually is right down here with this tRNA as well. This is actually easiest to see in a video, which I gave you a link to, but you can actually see here in a much easier process. We're starting out with the ribosome with an E site tRNA, a P site tRNA, and now what we're going to see is the process where we have EFTU together with the aminoacyl tRNA coming into the A site. The messenger RNA here is in gray. You can also see that it's really pretty bent, actually, in fact, not unlike the templates that you have. And there are these interactions between the codon and anticodon here, but also interactions between EFTU and the tRNA. So now we've had the appropriate interaction, EFTU and the tRNA, and the tRNA is the correct tRNA, has this beta loop structure next to the tRNA. That then binds to the 16S, the small subunit ribosomal RNA, wobbles around, and this wobbling is saying, hey, we're in the right position. Now we're in the right position. Now we have removal of this inhibitory region because it's interacted here properly. This then hydrolyzes the GTP. Once the GTP has been hydrolyzed, then the EFTU is dissociated, basically peeled off of this tRNA. That end, the piece that you just saw there, that's where that amino acid is. That gets brought to the peptidyl transfer site, and you have this transpeptidation reaction that takes place. And then, of course, you have the next step going on through elongation, etc. Still amazes me at the geometry of all of these things and how they all fit together appropriately with each other. So we've talked about initiation, we've talked about elongation, now termination. So termination happens when you hit a stop codon. And those stop codons, pardon me, are here. UAG, for instance. There's no tRNA that can now bind to this. What happens, instead of a tRNA, one of these RFs, release factors, binds to this A site. And once this RF is bound to the A site, and we'll see the molecular aspects of this in just a second, that now causes this terminal amino acid to actually form a peptide bond to water. Now this peptide is released. Now you have translocation that happens. This tRNA that used to have this attached to it just thinks that it's just transferred it to another tRNA which is here, but it has no idea. It's actually moved to the exit site. So now in the exit site, that can be released. The translocation happens, and then the whole thing falls apart. And this is really, again, pretty analogous to what you see with RNA polymerases. As soon as the RNA is removed from the RNA polymerase, everything falls apart. As soon as the protein is removed from the ribosome, everything falls apart. As it turns out, in this case, you also need some extra proteins to help pull the whole process apart. But let's first look at where these release factors are. This is a structure of one of those release factors, interacts with a messenger RNA right here, has, instead of a amino acid at this end, 3 amino acids, G, glutamine, so glycine, glycine, glutamine at the end. And this is where otherwise this peptide, which is attached to the P site RNA, would attach, but there's nothing to attach to there. This has actually also been found in high-resolution structures. Here we have just the structure itself. Here's a tRNA. Here's one of the release factors. Says it's about the same shape. I have trouble believing it's actually the same shape. Since this was published in our textbook, a more updated structure has been found where we now have a release factor here associated with the ribosome. tRNA is in the E site and the P site. This is now that mimic. Again, doesn't look too much like a tRNA to me, but still has this GGQ sequence right here, right at the catalytic site, which is where you have transpeptidation to water and release of the protein, which then, of course, leads to dissociation of the ribosome. But we have a couple of other proteins that are involved in this process. First is just that one that we looked at. Release factor 1 with the glycine, glycine, glutamine, binds to the ribosome, causes dissociation of the peptide. Now this peptide is dissociated. Turns out that this structure actually doesn't fall apart by itself. It needs help. And partly that's because you don't have a nice, translocation process whereby this protein can be moved into the P site. It's a different protein. So it turns out you have other release factors. In bacteria, this is release factor 3 that will bind to release factor 1, again bound to a guanosine nucleotide diphosphate now. Once this binds, that allows dissociation of RF1, but only if you get GTP bound to RF3. GTP bound RF3 can then hydrolyze its GTP to GDP and change its structure. This can be released. Now you have a ribosome that has 2 RNAs, tRNAs associated with it in the E site and the P site, but the peptide is long gone. So then we have the activity of, again, EFG, GTP, so the regular translocation process, only now the translocation process gives you E site no longer bound, the P site moves into the E site. This then dissociates these processes, and you get association of IF3. So what the heck is IF3, an initiation factor, doing here at the termination step? Well, what that initiation factor is doing is blocking these two guys from coming back together. So as soon as you have the dissociation again through EFG-GTP, put on IF3 stops the two of them from coming back together until they can start and do another round of translation. Find a new ribosome binding site or find a new cap, as the case may be, depending on if you are a eukaryotic or a bacterial translation machinery. Eukaryotic machineries, as we've talked about before, these are circular messenger RNAs because of EIF4G that forms a bridge between the cap binding protein, EIF4G, and the poly A binding protein. Your messenger RNA has an AUG on it. This will be elongated, but actually it turns out that there are usually multiple ribosomes bound to any messenger RNA. And as soon as the ribosome gets to the stop codon here, these guys dissociate, but they're right next to the 5' end. And so they can reassociate with the cap structure, translocate, reassociate, and go through the process again. So this looped structure really helps with getting lots of translation from messenger RNAs. And it's also called a polysome. Again, a horrible name. But polysome should really be polyribosome, where you have multiple ribosomes. You can actually see this. You can isolate what are called polysomes from cells that are actively translating messenger RNAs. So it's a messenger RNA with lots of ribosomes associated with it. And you look at an electron microscope, this is what you see. So there's your RNA with multiple ribosomes. Each of these guys is a ribosome that's associated with that one RNA. So I'd like to finish up today's lecture by talking about a couple of different things that happen as far as translation is concerned. When I talked about the genetic code, I mentioned that we have 20 amino acids. I said most cases we have 20 amino acids. There are actually a couple of extra amino acids, and one in particular is one called selenocysteine. Now, what the heck is selenocysteine? Well, selenocysteine is a amino acid that instead of sulfur in it has selenium. Turns out that selenium is a very important micronutrient. We all need selenium. In fact, present in most of our diets, but if you need more, you're present in vitamins as well. That selenocysteine actually gets added by a very specific tRNA. And this tRNA has the anticodon UCA. Okay, so what the heck is UCA? UCA can actually base pair with a stop codon, UGA. So this is the process and how you have charging of selenocysteine onto a tRNA. Actually happens through a serine, and that serine gets modified to selenocysteine by some very specific enzymes. Then this tRNA with selenocysteine on it binds to a very specific separate translation factor. This translation factor only works with selenocysteine. If you have selenocysteine and this translation factor, it can bind to a stop codon, but also only if you have yet another signal, usually this is in RNA, that now also associates with this translation factor. So if you didn't have this extra signal, anytime you had a stop codon and selenocysteine around, because we've got selenocysteine around as something that we need in many of our cells, that then would be incorporated every time you had a stop codon, and this would be bad. So here we have a secondary structure in the RNA, which allows this translation factor to associate with the selenocysteine tRNA and get a recoding, i.e. this codon, which is normally a stop codon, you have selenocysteine that's added here. Once selenocysteine is added, this is a totally normal peptide bond here. So this can now continue, and you've moved past this particular stop codon, because that stop codon is actually being used as a selenocysteine. So this is one way that you can end up with a messenger RNA that you think would stop, but it turns out actually adds an extra amino acid. There's also this process called frameshifting. So you remember our open reading frames, right? +1, start nucleotide 1, 2, 3, 4, 5, 6, etc., moving along through your messenger RNA. In some cases, and this is not just in viruses, but it's best known in viruses, is there'll be, again, a secondary structure which forms in the RNA. That secondary structure in the RNA actually fools the ribosome into copying different nucleotides through the presence of the amino acyl tRNA. So in this case, if you have no frame shift, no change of frame, you have UUU, UAA, GGG. But then if you have a frame shift, and very often these frame shifts are in areas where you have whole runs of U's, or actually for that matter, any particular nucleotide, this UUU is phenylalanine And then UUA is leucine. But now, instead of getting glycine here, now you have AGG, which encodes an arginine. And what that does is it changes the reading frame. And in the case of these viral genomes, that gives you a much longer protein, because now the stop codon that was otherwise here is not in the same frame. So it completely reads past this whole process here. I want to talk about quality control, or do I want to talk about that next time? Yeah, let's talk about quality control now, and then we'll start talking about protein folding next time. So, why do you need quality control? Why do you have to have or make sure that ribosomes are translating the right messenger RNA? And if you've got problems with the RNA, how do you deal with that messenger RNA? Part of the reason for that is, as I've mentioned quite a few times already, translation is an incredibly energy-using part of the whole process of going from DNA going to RNA for protein. And the reason for that is that for every amino acid incorporated, 4 or at least 4 nucleotide triphosphates have to be hydrolyzed. And that process is you have 2 ATPs that are going to be hydrolyzed to make your tRNA for the charging process. And for elongation, you have your GTP, which you have hydrolyzing for translocation, and the GTP that you're hydrolyzing with EFTU to make sure that you're getting the appropriate amino acid. And you have polysomes, so that means that on any given RNA, this could either be bacterial RNA because they're all lining up right next to your RNA polymerase, or your looped RNAs between the 5' end and the 3' end of your messenger RNA. These now are multiple ribosomes going down one messenger RNA. So if there's a problem with that messenger RNA, you want to stop translating because it's going to save you some energy, and you probably need those ribosomes for something else. So, couple of things that can happen to messenger RNAs. The first one is you have a broken messenger RNA, i.e. a messenger RNA that doesn't get to the stop codon. And if it doesn't get to the stop codon, as we talked about already with all the release factors, the ribosome is just going to sit there because there's no way that it knows to stop and dissociate. So, how is that dealt with? One of the ways that it's dealt with in terms of eukaryotes, since you've got 5' and 3' ends interacting with each other, there's a lot less opportunity for these breaks to happen. The ends are protected. But that's not true in bacteria. So in bacteria, you have this protein, actually it's a messenger RNA that's kind of like a tRNA, also called tmRNA. The name for it, the gene, is SSRA. So what does this tmRNA do? And it works a lot like a tRNA and a messenger RNA. What it does is it basically puts what I like to call the eat me tag, quote unquote, on the end of a protein saying, this is a bad protein, it needs to be degraded. So that's shown here, where we have a ribosome that gets stuck. There's a broken messenger RNA, again, probably through to some kind of exonuclease activity. This empty A site that doesn't even have a codon in it. There, so you have the tmRNA. So the tmRNA has a part that's like a tRNA that adds an alanine to it. And then it's got a part which is like an mRNA, which is right next to it. So what happens here is that this tmRNA binds in the A site where you don't have a messenger RNA. And then this messenger RNA, together with the ribosome, forms a peptide bond, this alanine. And then you have translocation of the ribosome. This broken messenger RNA gets out of the ribosome, gets degraded because the exonuclease is inside the cell. The tRNA that was here is now on the E site. That will get lost. And now you have the tmRNA in the P site. tmRNA in the P site, alanine. Now this part encodes for that tag, the eat me tag. Now this is the part of the tmRNA that's pretending to be messenger RNA. So here you have a regular tRNA, charged tRNA, brings in this case an alanine, that forms a peptide bond, and so on and so forth, ends up giving you this 11 amino acid tag at the end of your protein sequence that says, hey, this part of the protein is probably incomplete, we don't want it, so let's get rid of the whole thing. The other great thing about this is it's got a regular stop codon at the end. That means the ribosome can be released. It can go off and find, hopefully, a non-broken messenger RNA in order to translate. This is what happens in bacteria. tmRNA is only present in bacteria. Eukaryotic systems, there are other control mechanisms. So, as hopefully we all remember from talking about messenger RNA processing, one of the things that happens in messenger RNA processing is you have splicing. After you have splicing, then you form these exon junction complexes. Exon junction complexes is, hey, splicing has happened, we're good. In normal eukaryotic translation, as the ribosome moves through here, these exon junction complexes are removed, and you end up getting to your stop codon. You've made the protein, that you were looking for. On the other hand, if you have a premature stop codon, also known as a nonsense codon, so nonsense is stop codon because it doesn't code for amino acid, before you actually get to where you're supposed to be done at the normal end of your messenger RNA, you're going to have some exon junction complexes that are still associated with your messenger RNA because they haven't gotten bumped off by the process of the ribosome. That ribosome has gotten stuck. It's gotten stuck right here. So what happens in this case is that there are other proteins associated with the ribosome which then say, hey, there's an exon junction complex associated with this messenger RNA. There shouldn't be. So that means something is wrong. So now get rid of this cap structure. Get rid of the cap structure that is no longer protecting the end of your messenger RNA. It also doesn't form a bridge anymore to the 3' end of your RNA. So this gets chewed up by exo and endonucleases, which are going to chew up this uncapped, unprotected RNA because the ribosome ended up in the wrong place. So this is what's called nonsense-mediated decay. You can have exactly the same problem that's going to happen if you've somehow mutated your normal stop codon. And so this mutated normal stop codon, what happens now is instead of having a stop codon, the ribosome is going to keep translating your messenger RNA and get to the poly A tail. Once it gets to the poly A tail, it's going to put in a whole bunch of lysines. All these lysines because that's what translation of AAA is. AAA is UUU, which is lysine. So you end up with all of these lysines ended up at the end of your protein. This is also kind of an eat me signal that says, hey, this is a bad protein. The other thing that happens is once you get to the very end here in these poly A tails, you're bumping off the poly A binding protein. And so if you bumped off the poly A binding protein by the ribosome moving through it, then you activate this protein called the exosome. We already looked at the exosome. This breaks down RNAs in the nucleus, but there's also exosomes that are present inside the cytoplasm of cells. Those exosomes can then also chew up your messenger RNA. And so getting to the end, no more poly A tail, no more poly A binding protein because the ribosome's gotten rid of it. Now the exosome can chew up your messenger RNA. So the last clicker question, and actually the end for today, presence of which of the following on messenger RNA leads to nonsense-mediated decay? Cap binding complex, EF4E, EF4G, exon junction complexes, or EIF2. Nonsense. What does nonsense mean? Nonsense means stop codon, and a stop codon in the wrong place. The way that that's recognized is not by where the cap is binding, because that's going to be binding at the 5' end. EF4E is also part of this cap binding complex. EF4G is forming the bridge. EF2 is important in initiation, getting your initiator tRNA. It's got to be exon junction complexes, and that's actually just like what we talked about. Nonsense. What does nonsense mean? Nonsense means stop codon, and a stop codon in the wrong place. The way that that's recognized is not by where the cap is binding, because that's going to be binding at the 5' end. EF4E is also part of this cap binding complex. EF4G is forming the bridge. EF2 is important in initiation, getting your initiator tRNA. It's got to be exon junction complexes, and that's actually just like what we talked about. So we'll stop here. We'll talk about antibiotics and protein folding on Friday, hopefully in person. If not, stay safe and don't slide around too much.